Hello everyone, welcome to another video session. Today we will understand and learn the process of endochondral ossification. So this is a continuation of my previous video which was based on the intramembranous type of ossification. So the basics of osteogenesis have been covered once again for your understanding, and then we'll get into the detail of endochondral ossification. The term osteogenesis is derived from two words, that is osteo meaning bone and genesis meaning formation. Therefore, osteogenesis is the process of bone formation. It usually begins between 6th and 7th week of embryonic development and it continues till around 25 years of age. If we have a look at this process embryologically, it is the cranial neural crest cells which form the flat bones of the skull, clavicle and the cranial bones, excluding a portion of the temporal and occipital bones. The somites form the remainder of the axial skeleton, which include the skull, ears, neck, back and the ribcage, while it is the lateral plate mesoderm which forms the long bones. The process of bone formation requires a template for development, and this template is usually in form of a cartilage derived from the embryonic mesoderm, but also it includes an undifferentiated mesenchyme. So based on this, the bone ossification can either be intramembranous or endochondral. So each of this process begins with the mesenchymal tissue precursor, but it differs in how this precursor is transformed into bone. Intramembranous ossification directly converts the mesenchymal tissue into bone and forms the flat bones of the skull, clavicle, and most of the cranial bones. Endochondral ossification, on the other hand, begins with the mesenchymal tissue, which transforms into a cartilage intermediate, and it is this cartilage intermediate which is eventually replaced by bone. This ossification is responsible for the formation of remainder of the axial skeleton and the long bones. So I found this illustration on web where they differentiate into the sites of ossification. For the intramembranous ossification, they use this memory aid that the skull needs to ossify fast, considering that the skull bones must ossify prior to the delivery of the fetus, so the brain isn't squashed during childbirth, because intramembranous type is more of a direct form of ossification. While for the endochondral type, they use this mnemonic, that is kids better bounce not break, because kids, they grow extensively during the pediatric development and therefore their bones require some amount of pliability which is offered only through the cartilage. Therefore, considering that for that particular time the children fall often and therefore they must bounce and not break. As mentioned previously, both the kinds of ossification begin with the mesenchymal derivative. So in endochondral type of ossification, this derivative differentiates into chondrocytes as compared to osteoblast, which was happening in the intramembranous ossification. These chondrocytes then proliferate rapidly and secrete an extracellular matrix around them, which forms the cartilage model for the bone. This cartilage model includes the hyaline cartilage and it resembles the shape of the future bone, and it is also surrounded by a membrane called the perichondrium. Peri because it is surrounding the chondrium, that is the cartilage. After this, the chondrocytes that are present at the center of the bony model begin to undergo hypertrophy, which means they start increasing in size. Along with this, they also start adding collagen X and more fibronectin to the matrix that is being produced. This altered matrix allows for calcification. Calcification of the extracellular matrix that happens prevents the nutrients from reaching the chondrocytes and ultimately causes them to undergo apoptosis or cell death. This leads to creation of voids in the cartilage template and allows for the blood vessels to invade. These blood vessels enlarge the spaces and eventually combine and become the medullary cavity at the center. So the blood vessels also carry with them some osteogenic or bone forming cells, and this triggers the transformation of the perichondrium to periosteum. . The middle region of the long bone, also known as the shaft, is the diaphysis of the bone, and in this region the osteoblasts create a thickened region of compact bone, also called the periosteal collar. So it is this area in which the primary ossification center forms. So the long bone can be divided into three areas: the middle diaphysis, the end of the bone which is the epiphysis, and the area in between the two which is the metaphysis. So while the bone is being replaced by cartilage in the area of diaphysis, at the end the cartilage continues to proliferate in order to increase its length. So these proliferative areas are also known as the epiphyseal plates, the physeal plates, all the growth plates, and these allow for the longitudinal growth of bones to occur after birth and also into the early adulthood. Now let's have a look at the zones of this growth plate. It lies between the epiphyseal and the metaphyseal bone, and it has three zones mainly. The first is the resting or germinal zone, and it contains stem cells or pluripotent cells. The second is the proliferative zone, which contains proliferating chondrocytes and pre-hypertrophic chondrocytes. And the third is the hypertrophic zones, which as the name suggests contains hypertrophic chondrocytes. This entire process of formation of primary ossification center at the diaphysis repeats itself after birth in the epiphyseal region at the ends of the bone, and this is where the secondary ossification centers form. Now let's revise and summarize this entire process. So the endochondral ossification begins with the mesoderm, in which the mesenchymal cells migrate and differentiate into chondrocytes, which secrete an extracellular matrix. This leads to formation of a hyaline cartilage model, at the center of which the chondrocytes begin to undergo hypertrophy and there is calcification of the matrix. After this, there is invasion of the blood vessels into the voids of the model, and this is followed by the formation of medullary cavity at the center, formation of a periosteal collar, and transformation of perichondrium to periosteum. And there is formation of primary ossification center at the diaphyseal area of the bone. So while at the middle the cartilage is being transformed into bone, at the end the cartilage undergoes proliferation because of the formation of growth plates. So this entire process repeats itself after birth at the epiphyseal ends of the bone and leads to the formation of secondary ossification centers. So that was all about the process of endochondral ossification. I really hope that with the help of the diagrams and the illustration you were able to grasp the concept. For any further doubts, you are free to drop the comments in the comment section below.